Hi, it's Dr. Blossman, and we'll be discussing gluconeogenesis in this video. Gluconeogenesis is the production of glucose from a non-carbohydrate precursor, maybe pyruvate, that was produced in glycolysis and will not be used for aerobic respiration. Maybe the cell needs more glucose, so it will activate gluconeogenesis, and at the same time it will inhibit glycolysis. So let's go through these steps. Again, gluconeogenesis is making glucose from pyruvate, and it's not the exact reversal of glycolysis. It has three reactions that differ from glycolysis. Remember, glycolysis is 10 reactions with 3 points of control. Well, this will be the flip of that pathway, but again with some exceptions. So it's not a strict reversal. There were three control steps in glycolysis. The first one, the last step 10 was phosphoenol pyruvate, which is also known as PEP. When it was converted to pyruvate by pyruvate kinase, that is going to be controlled. Fructose 6-phosphate to fructose 1,6-bisphosphate. This is the most important control step of glycolysis. This was in step 3, and this was catalyzed by PFK1, which is an allosteric enzyme that's also controlled by covalent modifications. And then the step 1, glucose to glucose 6-phosphate, these were the 3 points of control steps. In this instance, in my PowerPoint, I have them backwards. So they go from, this is step 10, this is step 3, and this is step 1. These are points of control. Remember, this step, catalyzed by PFK1, was marrying the glucose into glycolysis, meaning that once it's there, once it's become a fructose 1,6-bisphosphate, it's committed to undergoing the entire metabolic pathway. But the net result is going to be a reversal of glycolysis, gluconeogenesis is, but we're going to discuss how it differs. This is a very helpful chart. Let's just break this down over here on the red side. This is glycolysis, and as you can see, I have put a box around the reactions that are different between glycolysis and gluconeogenesis. So we're going to be differing in step 1 of glycolysis, we're going to differ in step 3 of glycolysis, and then again in step 10 of glycolysis, which will be the first reactions of gluconeogenesis. We're going to actually split those into 2 reactions, and of course it's differing. We are in steps 1, 3, and 10, which happen to be the control steps of glycolysis, is where we will differ with gluconeogenesis. So not essentially the reverse, but a lot of the reactions are just, strict the other way. Okay, so quick review of glycolysis. We have 10 steps from glucose to pyruvate. So on the blue side over here, this is going to be gluconeogenesis. But I will note that glycolysis was done in the cytosol of the cell, and gluconeogenesis is going to be in 3 different locations depending upon the reactions. So here, steps 1 and 2 will be done in the mitochondria, with a caveat that step 2 can be done in the mitochondrial matrix or in the cytosol, because there is an enzyme, the PEP carboxyl kinase, or PEP-CK, will be in the mitochondrial matrix as well as the cytosol. So that one's not really cut and dry where that reaction will take place. Make sure that you know that pyruvate carboxylase will definitely be in the mitochondrial matrix. So step 1, we're going to be converting pyruvate to oxaloacetate. Oxaloacetate you might recognize as one of the first starting intermediates of the Krebs cycle or the CAC cycle. And this, again, is a 2-part reaction to go from pyruvate to oxaloacetate catalyzed by pyruvate carboxylate. The next reaction is going to catalyze oxaloacetate to something that should be familiar, the PEP phosphoenolpyruvate, which is going to be the same for glycolysis, the same intermediate. PEPCK is going to be the enzyme again that is located in both the mitochondrial matrix as well as the cytosol. So this second step can be done in the mitochondrial matrix or the cytosol. In either case, it takes two reactions to get from pyruvate to phosphoenolpyruvate. Okay, steps 3, 4, 5, 6, 7, and 8 of gluconeogenesis are the essential reverse reactions of glycolysis. Remember, in step 7 is when we created an ATP. Well, in step 5 of gluconeogenesis here, that's when we're going to be using one. Okay, so again, this is a reverse. We're going to go from the 3 carbon molecules, the glyceraldehyde 3-phosphates, we're going to put them together, back together again, in the reverse reaction of aldolase. So here, step 9 of gluconeogenesis, we start with fructose 1,6-bisphosphate, and this reaction is not catalyzed by PFK, so it's not, PFK was not a reversible reaction, it was going one direction, but we have an enzyme, fructose 1,6-bisphosphatase. Okay, so step 9, fructose 1,6-bisphosphate is going to be catalyzed by fructose 1,6-bisphosphatase. The reaction is going to end up with fructose 6-phosphate, which should look familiar from glycolysis. And so it's the same intermediate, just different enzyme catalyzing the reaction because PFK1 is not a reversible reaction. Then we have the next step, which was just the isomerization, which is the same but reverse. And then the last step, which was catalyzed by hexokinase in glycolysis, now we're going to use glucose 6-phosphatase to remove a phosphate. When you have a kinase, you're putting on a phosphate. When you have a phosphatase, you're taking off or you're dephosphorylating. So that should make sense. A kinase would be putting on the phosphate, going in this direction in glycolysis, and a phosphatase is taking it off and creating glucose. This occurs in the endoplasmic reticulum. So step 11 of gluconeogenesis is in the endoplasmic reticulum. Steps 3 through 10, or potentially 2 through 10, are occurring in the cytosol, with definitely step 1 of gluconeogenesis occurring in the mitochondrial matrix. So we have a division of 3 different locations for the reactions of gluconeogenesis. Whereas with glycolysis, we could say that all the enzymes are in the cytosol, but for gluconeogenesis, the enzymes for gluconeogenesis will be located in the mitochondrial matrix, in the cytosol, as well as the endoplasmic reticulum for the final step. Okay, looking at the chemistry of it pretty briefly, we have pyruvate, and this is going to be a carboxylation step to oxaloacetate. Whenever we have a carboxylation step, we need biotin, which is vitamin B7, which will act as a coenzyme for pyruvate carboxylase. Biotin is going to put that carboxylate group on to whatever it's reacting with. So in this case, we have pyruvate becoming carboxylated by a carboxylase that has a coenzyme of biotin. Usually magnesium is going to be a cofactor. It does require ATP. Remember, in the reverse step in glycolysis, we synthesize the ATP, so it makes sense that if we go the reverse that we would use ATP. So we're creating the 4-carbon molecule known as oxaloacetate, which is a very popular intermediate. Not only is it used in gluconeogenesis, but it's used in the Krebs cycle as the first step, as well as other reactions such as creating NADPH in the cytosol for lipid biosynthesis, and so forth. So here's our ADP plus PI, inorganic phosphate. This is going to be a broken down ATP and then plus 2 hydrogens. So that's the first step. Again, this requires biotin, which is a carrier of CO2. Acetyl-CoA is going to be an allosteric effector for pyruvate carboxylase, and this will be inhibited by ADP and glucose. Think about what this pathway is. It's a production of glucose from pyruvate. So in the case where we have a lot of glucose, are we going to need to create more glucose? No. So it would make sense that here this pathway is going to be inhibited by glucose. It's also inhibited by ADP. Again, ADP would be one of the products of it, and it would be an indicator of not that much energy. Whereas ATP is showing you that we have a lot of energy available, whereas ADP is an indicator of low energy in the cell. Acetyl-CoA is going to be an allosteric activator for this enzyme. So if we have a lot of acetyl-CoA, then more than is necessary to supply the CAC, then that is going to notify the cell via using this as an activator to create more glucose from pyruvate. Oxaloacetate can also be a starting point for gluconeogenesis as well. And here I'm just showing you in a horribly drawn thing, where this CO2 is going to complex with biotin, which is the coenzyme on the enzyme pyruvate carboxylase, and it becomes a carboxylate, creating oxaloacetate. Okay, the next step, decarboxylation of oxaloacetate, is going to be coupled with the phosphorylation by GTP to give PEP, phosphoenolpyruvate. This here I'm showing you, we're taking this carboxylate group off. We're going to liberate it as CO2. And we're going to break down, we're going to break one of these anhydride bonds and use the energy for this. It's a coupled reaction and create the most high energy molecule in the body, phosphoenolpyruvate. The enzyme is phosphoenolpyruvate carboxyl kinase, and this is going to be using magnesium as a cofactor. Oxaloacetate at this point will go to the cytosol, but it does have a malate shuttle, another time, another place, but just know oxaloacetate proper does not cross the mitochondrial membrane. It's going to undergo a redox reaction and move across as malate. And here oxaloacetate says, I hate it that I can't leave the mitochondria. I have to put on a malate costume, but I feel so reduced when I do that. So oxaloacetate becomes reduced to malate, L-malate specifically, and goes to the cytosol. And then it becomes oxidized once it gets to the cytosol to oxaloacetate. Okay, so then, we had those reactions that were the same, basically the reverse of glycolysis. Then we get to this reaction, fructose 1,6-bisphosphate to fructose 6-phosphate. This is going to be catalyzed by fructose 1,6-bisphosphatase. Remember, a phosphatase is what removes a phosphate group. So here we're left with fructose 6-phosphate. This point is controlled. The allosteric enzyme is strongly inhibited by AMP and stimulated by ATP. So ATP, when we have it in high concentration, is an indicator of a lot of energy for the cell. This again is going to be in the cytoplasm. And then the last reaction that differed was catalyzed by glucose 6-phosphatase, another phosphatase occurring in the ER. This is where we take the phosphate off of the C6 of glucose 6-phosphate and we have glucose. And that's it for gluconeogenesis.